Cells don't act alone. They're constantly sending and receiving signals, and these signals control everything a cell does, like growth, differentiation, and even apoptosis, which is programmed cell death. In this video, we'll explore why cells communicate, how these signals travel from reception to transduction to response, and also cover the key vocab that you'll need for the AP exam or even your test. Communication lets cells coordinate activities like growth, secretion of molecules, and apoptosis. Homeostasis, which is the maintenance of stable internal conditions, depends on these signals. If cells can't communicate, the body can't function as a coordinated system. Only cells with the correct receptor respond to a signal. Even the same ligand can have different effects depending on the cell type. For example, adrenaline makes liver cells release glucose but speeds up heart contractions in cardiac cells. This specificity ensures each cell reacts appropriately. There are several ways cells communicate, but there are four major. Autocrine signaling affects a cell that released the signal. Paracrine affects nearby cells. Juxtacrine requires direct contact between cells, and endocrine involves hormones traveling long distances, typically through the blood, to target cells. A ligand is any signaling molecule that triggers a response in a cell. A receptor is the protein that detects the ligand. When the ligand binds, it causes a conformational change, which is just a structural shift in the receptor that activates the signaling pathway. Some ligands, like steroid hormones for example, enter the cell and bind intracellular receptors, which often change gene expression, the process by which DNA instructions are used to make proteins. Polar ligands, on the other hand, like protein hormones, bind extracellular receptors and trigger processes such as exocytosis. Signal transduction is the process by which a signal is relayed from a receptor to the cell's response machinery. It has three main steps: reception, where the ligand binds to the receptor; transduction, where the signal is passed down and often amplified; and response, where the cell does something. Amplification ensures that even a small signal can produce a large effect. Reception occurs when the ligand binds to a receptor and causes a conformational change, a shift in the receptor that activates it. A common type is the GPCR, or G protein coupled receptor, which activates downstream proteins in the cell. During transduction, the signal moves through a signaling cascade, passing through multiple proteins. Each step often amplifies the signal, allowing the cell to respond strongly even to a small initial signal. Many cascades involve phosphorylation, where kinases add phosphate groups to proteins, which activates them. Phosphatases remove these proteins to turn the signal off, and these phosphorylation cascades help amplify signals inside the cell. Often, secondary messengers like CAMP and calcium ions also carry the signal from the receptor to other parts of the cell. These messengers allow rapid and widespread activation of cellular responses. The cellular response itself can involve turning enzymes on or off, changing gene expression, altering behavior like movement or secretion, or even initiating apoptosis. This ensures cells respond appropriately to the signals they receive. Signal termination stops the response once it's no longer needed. Ligands are removed, secondary messengers are broken down, and proteins undergo dephosphorylation, which turns them off, which allows the cell to reset. Here's the classic G protein pathway. When a ligand binds to a GPCR over here, it activates a G protein, and this part of the protein, right here, the alpha subunit, moves to adenylate cyclase. AC then converts ATP into CAMP, a second messenger that spreads the signal inside the cell and activates downstream responses. This mechanism shows how a small signal outside the cell can trigger a large internal effect, a key concept for bio. Here are two questions for you. Let me read out the first question. Certain signal transduction pathways regulate whether stem cells remain as stem cells or differentiate into specific cell types. One such pathway, known to be important in regulating development in flies, is summarized in the diagram above, with pathway components defined in the diagram legend. A signaling polypeptide called DPP is found to inhibit the transcription factor GATA in the signaling pathway. Based on the diagram, it is most likely that increased levels of DPP would A. Cause stem cells to remain unchanged, B. Stimulate stem cells to differentiate, C. Induce transcription of the DFOG gene, or D. Reduce phosphorylation of STAT. Please pause the video here and come back when you have an answer. The answer is C. It stimulates cells to differentiate. The transcription factor GTA normally induces the transcription of DFOG, which blocks the differentiation, as seen by this over here. The question says that if GTTA is inhibited by the signaling polypeptide DPP, the inhibition would actually prevent GATA from inducing the transcription to prevent differentiation. As a result, the differentiation will be stimulated when DPP is present at increased levels. Here's another question for you. Describe the 3 stages of cell signaling: reception, transduction, and response. Please pause the video again and come back when you have an answer. Here's what your answer should sound like. Reception. It is the signal molecule where a ligand binds to a specific receptor in or on the cell. Transduction. It is a series of steps that relays and amplifies the signal inside the cell, and it often involves second messengers. Response is when the cell carries out a specific action, like gene expression or enzyme activation. Here are the key takeaways. Signals are specific. Only cells with the right receptor respond. Ligand binds these receptors, restarting the pathway. Signal transduction amplifies the message through cascades and secondary messengers like CAMP and calcium ions. Cellular responses vary, activating enzymes, changing gene expression, secreting molecules, or initiating apoptosis. Signals must terminate through dephosphorylation and ligand movements so cells can reset and respond to new signals through homeostasis. That wraps up our review of cell communication. Next, we'll cover the cell cycle, checkpoints, and how signals control cell division. Review all pathways, ligands, and receptors. They are the foundation for Unit 4 questions. Thank you.